Today we are going to talk about the cellular events during acute inflammation. In the previous lecture, we have already discussed how neutrophils are marginated from the central flow to the peripheral flow due to hemodynamic changes in the microcirculation of inflamed area. Today we are going to concentrate that once neutrophils come to the margin, right, and they become near the endothelial cells, they should attach or become adherent to the endothelial cell so that they are not taken away by blood flow out of the inflamed area, right? So what really happens that phenotype of these endothelial cells alter. Normally, endothelial cells in healthy tissue are not adhesive or sticky for the neutrophils. But when the inflammation occurs, I will explain the molecular mechanism later, but during the inflammation in post-capillary venules, the endothelial lining becomes pro-adhesive, right? It develops either adherent molecules or their ligands so that there is interaction between the endothelial cells and the neutrophils and they stick to each other, right? Now, how exactly this process occurs? First part is rolling. What is meant by rolling? Rolling is the first part in which neutrophils which are marginated initially they attach with the endothelial cells weakly. They attach with the endothelial cells loosely. They just attach and then blood flow will push them. So they will roll over. It means they were weakly attaching, so they detached and rolled. Then somewhere else in the endothelium, they will attach, right, weakly, and then again, due to pressure of the blood flow, they will again roll over. So what these leukocytes will be doing in early part of inflammation, that these leukocytes, or we call them more specifically neutrophils, so these neutrophils will attach, then detach, roll, then attach and detach and then roll. I will explain molecular levels later, but this weak attachment is provided or mediated by group of molecules called selectins, right? This weak attachment between the endothelial cells and neutrophil, this is mediated by group of molecules called selectins, right? So selectin mediated rolling of neutrophils will occur. Now, selectin mediated rolling of neutrophil is like neutrophil come, kiss the adhesive endothelium and then roll over, then again kiss, then move on, again kiss, then move on. So what's going on there? The love affair between the neutrophils, the love affair between the neutrophil and endothelial cell in the beginning is like rolling. Neutrophils are rolling on endothelial lining, right? Because they are attaching there, then they detach and roll on. Because this process repeatedly occurs, it is just like kissing and moving on. Again kissing, kiss and move on. Again kissing and moving on. So I will explain molecular levels in detail in few minutes, but just we think that this process where neutrophils are marginated and then become near the endothelium, their molecular mechanisms interaction occurs between neutrophils and endothelial cells, which depends on group of molecules called selectins. And selectins and their ligands provide weak bond. They provide very loose bond. So it is just like soft kiss. So neutrophil softly kiss the endothelium, move on, kiss the endothelium, move on. I think neutrophil is lucky. Kiss another endothelial cell and move on. So this kissing and moving on is called rolling of leukocyte. I will explain the molecular mechanisms just in few minutes. Then, once this process is occurring, that neutrophils are rolling over endothelium, attaching, detaching, depending on expression of selectins, then what happens? Then another group of adhesion molecules appear other than selectin, and that second group of adhesion molecule is called integrins, and integrins provide relatively strong bonding. They provide relatively strong bonding between the neutrophil and endothelial cell. So if a neutrophil after little bit rolling expresses integrins and their ligands, if neutrophil and endothelial cell develop activated high affinity integrin molecule and their ligands, they will bind with each other tightly. We call it stable attachment, or we call it that leukocyte arrest, or we call it firm adhesion, right? So, or we can call it that neutrophil which was kissing and rolling, eventually is caught by interaction of integrins and their ligands, and now there is a tight hugging. So what we are saying, that in these mechanisms, previously we told that if normally what happens from arterial side, as blood is coming, neutrophils or white cells, leukocytes, come into microcirculation and they exit through the tissue into venous drainage. But when there is inflammation, when there is injury, we need neutrophils to move out of vascular compartment to the tissue and help us to remove or to contain the offending agent. For this purpose, first neutrophils were brought out of central flow to the peripheral margination. Then first group of adhesion interaction occurs, which is dependent on selectins, and that is a weak interaction, weak adhesion. So selectins just help the neutrophils to attach weakly and transiently with the endothelial cells and then roll over, right? This weak and transient attachment and then moving on, what is that? Rolling. But within some more time, another group of adhesion molecules are activated and come into action. What are those? Those are integrin molecules. Integrin molecules and their ligands also become activated and appear, and the interaction between integrins on the leukocyte and their receptors, right, on the endothelial cell, they act strongly and there is leukocyte arrest. No more rolling and this naughty business of kissing and moving, moving and rolling and kissing, this is stopped, and now neutrophil is arrested on one specific endothelial cell and bind there strongly. This is called what? This is called firm adhesion. So we say rolling was selectin dependent. I will explain later what are selectins and how do they work. Then firm adhesion is integrins dependent. Rolling is like kissing and moving, kissing and moving, kissing and moving. Firm adhesion is like strong hugging. They are having a tight hug. Then, once neutrophil is strongly attached with the endothelial cell, then this neutrophil will spread out. It will not remain circular or spherical. It will spread out. So blood flow does not push it too much. And this strongly adherent, firmly adherent, integrin dependent attachment of neutrophil also activate that neutrophils will spread out, and already kissing have been done, hugging is done. Now this neutrophil will spread out on the endothelial cell and look for some gap. It's a love affair. So what has happened? It is looking some crack or some gap where classically in between the two endothelial cells. Then neutrophil will penetrate through that towards the extravascular compartment. This is deep penetration of neutrophil in between the endothelial cell. We call it paracellular diapedicis. Movement of neutrophils from vascular compartment through the endothelial lining towards the extravascular compartment is called diapedicis, or it is called transmigration through the endothelium. Now, there might be deep penetration in between the two endothelial cells. Through this, neutrophil will try to slip away into extravascular compartment, and this is diapedicis, and it depends on a group of molecules. I will explain later in detail, but here I just mentioned these molecules are PCAMs, JAMs, and CD99. So it is deep penetration, but sometimes neutrophil do brutal penetration. What is brutal penetration? This love affair is not going well. Brutal penetration means that sometimes neutrophil pass through the endothelial cell. This diapedicis, which is through the endothelial cell, is called transcellular migration. Transcellular migration, right? Let me make the major thing, then we go to rolling in detail. Neutrophil came in the central flow. Hemodynamic changes pushed them on the lateral side or onto the peripheral side, called margination. Then endothelial cells and neutrophil express group of adhesion molecules called selectin. There is selectin mediated weak and transient bonding, and then detachment, then again attachment, then again detachment, kissing and moving, kissing and moving, that is rolling. While rolling is going on, the purpose of rolling is to slow down the movement of neutrophil towards the exit. Once neutrophil becomes slow down, then another group of adhesion molecules are expressed on the, what is this, endothelium, and on their counterparts appear on leukocyte. On the leukocyte, there are integrins which become very active, and here their ligands appear, and now neutrophil will strongly bind. It will no more move. We say there is neutrophil arrest. We say there is integrin dependent firm adhesion. Once it becomes adherent to one specific endothelial cell, then neutrophil will spread out and look where it can find some gap in between the two endothelial cells. Then through the gap, what is this? Neutrophil will start migrating towards the basement membrane and eventually extravascular compartment. This movement is called paracellular transmigration or paracellular diapedicis. But sometimes what happens that neutrophil pass through and through the endothelial cells. I will explain later exactly how it happens. And that is called transendothelial migration or diapedesis of neutrophil. Now, let's come into detail. Let's focus right now, focus on rolling. That from where the selectins come, from where their ligands or selectins receptors come, and how the attachment occurs. Let me tell you, when inflammation occurs in this tissue, right, in this tissue lot of chemical mediators are produced, right? When any tissue is injured, either by the pathogen or by the necrotic cells, our own necrotic cells, or due to some foreign body, some chemical injury, thermal injury, or any kind of injury, when tissue are injured, then lot of chemical mediators are produced. Some of the chemical mediators which can mediate the steps of inflammation, they are produced by mast cell. As you know, some are produced by resident what? Macrophages. Some mediators are produced by the injured endothelial cell, and still some chemical mediators of inflammation are produced by the plasma proteins, different kind of plasma protein. Let me explain first of all. Now, this step of rolling, this is initiated by two special chemical mediators of inflammation. I'm going to draw this endothelial cell here. This is the endothelial cell, and what are here? This is histamine going to act on it, histamine and thrombin. Thrombin is produced by fibroblast and also produced by endothelial cells themselves when endothelial cells are injured, right? And thrombin can come from the activation of certain coagulation pathways. But right now, what I want to say, that histamine and thrombin, they act on the endothelial cell. You know, source of histamine is mainly coming from mast cell. So at the site of inflammation, right, mast cell release histamine, and even if there is some thrombin, histamine and thrombin will act on endothelial cell. So when histamine and thrombin, right, here I am writing histamine, and histamine is coming from mast cells, and thrombin can come from blood product. Thrombin can come from the coagulation factor cascade pathway, or it can be released by injured endothelial cell. Both of them, when they act on endothelial cell, inside the endothelial cell, there are special granules. And these granules are very rich in especially von Willebrand factor, which we will not discuss now. Plus, in the membrane of this vesicle or granule, there are preformed selectin molecules, and those selectin molecules, they are found in the platelets, in the alpha granules, and also they are found in these granules in endothelial cells. They are pre-synthesized and preformed and stored in the membrane of this granule. Now this endothelial cell granule has many different products, but we will just talk about two most important. Number one, they have von Willebrand factor. Number two, in their membrane of the granule, they are preformed, pre-synthesized molecules of P selectins. What are these molecules of P selectins? Now, why we call it P selectins? Because originally they were discovered in alpha granules of platelets. But now we know they are also present in these granules. These special granules have a very special name. The name of this granule is, yes, Weibel plate, PLADE, plate bodies. So now we can say the endothelial cells normally are having lot of special granules called Weibel plate bodies. These granules are loaded with von Willebrand factor, and also in their membrane we are having preformed, pre-synthesized P-selectins, right? Why we call them P-selectin? Because they were originally discovered in alpha granules of platelets. But now we know P-selectin is a type of selectin which is present in platelets, alpha granules, and also present in endothelial cell in Weibel plate bodies. Now, when histamine activates this endothelial cell, or thrombin activates these endothelial cell, this activated endothelial cell will move Weibel plate bodies towards the luminal side, luminal face of the endothelial cell. And when this will move towards the lumen, right, its membrane will fuse there, and when this will fuse with the membrane, of course von Willebrand factor will be released, but P-selectins will become the part of the endothelial cell membrane. P-selectins will become the part of the endothelial membrane. In this way, we can say P-selectins will appear over here. P-selectins will appear here, right? Now, exactly what is the structure of P-selectin? Remember, there are three types of selectins. There are P-selectins, which are found in platelets and endothelial cells. There are E-selectins, which I will discuss later. E-selectins are specifically found more commonly in endothelial cell. So P into platelets and endothelial cell, E-selectins mainly on endothelial cell, and in the end I will talk about L-selectin. L-selectin are found on leukocyte membrane. Leukocyte membrane. So there are three types of selectins, right? Selectin is the smallest group of adhesion molecules: P-selectins, E-selectins, and L-selectins. P-selectins on, yes, platelets, in platelets granules, alpha granules, and P-selectins on von Willebrand factor. Now, because these P-selectins are preformed, right? Even though they are peptide, they are actually eventually they become transmembrane, single transmembrane glycoprotein. Single pass, they pass once through the membrane. So we call it single pass transmembrane glycoproteins. You know, synthesis of protein takes some time, but here they were already synthesized and stored. So as soon as injury occurs, then in this microcirculation, in these endothelial cells, within few seconds to minutes, they start expressing P-selectins. They start expressing P-selectins, right? Why? Because as soon as injury occurs, histamine is immediately released from, yes, from the granules of injured mast cells, and thrombin can be released from injured endothelial cells. So histamine and thrombin trigger the movement of Weibel plate bodies towards the luminal surface and insert the P-selectin in the membrane. And what are P-selectins? I will show it here, that P-selectins are basically, okay, I will show you here, right? This is endothelial cell and here is P-selectin. Now what is P-selectin? P-selectin has one transmembrane part, one cytoplasmic tail, this is cytoplasmic tail, and outermost area, there are some other part, but outermost this area of P-selectin, this area is lectin binding domain. What is this? Lectin binding. Lectin binding domain. And this binding is calcium dependent. Now, when P-selectin is inserted into membranes very rapidly at the onset of inflammation, P-selectins have outer extracellular domain which is lectin binding domain. Lectins basically carbohydrates. So it is carbohydrate binding domain. Carbohydrate binding domain means that they are going to attach with some sweet thing, right? Now, we can say by expression of P-selectins on endothelial cells, right, makes the endothelial cell pro-adhesive. But P-selectins are weak binding. But before binding, we should know there should be some corresponding receptor on neutrophil to bind the P-selectins. There should be corresponding. So what happened? It is just like that. These are the P-selectins are the lips of endothelial cell. But if neutrophils are going to kiss here, neutrophil should have corresponding lips. This is kissing, soft kissing. So, from endothelial cell, what lips are expressed? P-selectins. And from the neutrophil side, also corresponding adhere molecules will be expressed. And these molecules, I will explain in within 1 minute, but just concentrate here. Meanwhile, neutrophils are also activated by by chemical mediators of inflammation, and neutrophil also express special receptors, or we can say they express P-selectin ligands. They express P-selectin ligand. I will explain their structure also. But this is, for example, yes, this is coming from which side? Neutrophil membrane, right? This is coming from neutrophil membrane. Now neutrophil is very happy. Soft kissing is going on, right? I hope this should be happy too, only smiling. Okay. Now, P-selectin here, now what is this called? There are many different books mention it in different way. So I will tell different terms used for the same molecule or similar molecule so that you don't get confused. Some books will write that this is P-selectin glycoprotein ligand. This is going to bind with the P-selectin. So this can be called P-selectin glycoprotein ligand 1. Right? Basically, this is also glycoprotein, right? But it has very special, you can say, domain. I will mention later. But P-selectin glycoprotein ligand. P-selectin glycoprotein ligand 1. This is one name for this corresponding receptor. Then this has one more name, right? Let me explain this structure. This structure is basically this component of the structure. This is glycoprotein, right? This is neutrophil membrane, right? This is neutrophil membrane, right? Now, This structure up to here is called what? Glycoprotein. This is structurally glycoprotein, but this component, it is a very special component. Now I'm going to draw it in proper detail. This component is oligosaccharide. It is a short chain of monosaccharides, right? So we call it oligosaccharide. So here is oligosaccharide and there are, this is suppose one monosaccharide, then there is another monosaccharide here, and here is one more, and then one more. Now this is basically small chain of monosaccharides. So it cannot be called polysaccharide because it is not long chain, it is short chain. We call it oligosaccharide, right? Now a very important point, this one is sialic acid. Sialic acid, Sa is equal to sialic acid. So it means this receptor is glycoprotein along with oligopolysaccharide, oligopolysaccharide having a group called sialic acid, because here is your sialic acid, you can magnify it. Here is sialic acid. This was suppose carbohydrate number 1, this was another carbohydrate, and this is sialic acid. Now, so another name for this is, whole molecule is, in some other books, rather than they mention that this is p-selectin glycoprotein ligand 1, they simply call as a group. As a group, such molecules are called that these are sialated oligosaccharides attached on glycoproteins, right? Attached with mucin like glycoprotein. So another name for this is sialated oligopolysaccharides attached to glycoprotein. So this is, for example, in case of PSGL1, there is glycoprotein which is attached with the neutrophil, and with that there is oligosaccharide which is sialated. So we also call it sialated oligosaccharides attached. Okay, don't write polysaccharide, it is oligosaccharide. Sialated oligosaccharides attached to glycoprotein, and this group act as receptor or counter molecule for the p-selectin. Now the good news is that E-selectin also bind with the same molecule. So it means PSGL1 act as a receptor for p-selectins, also act as a receptor for E-selectins. What are E-selectins and how they are produced, I will explain in a few minutes. But now another point. This point is very important. I will explain later. Now, this is sialated part. Another name for this black area, this oligosaccharide which is sialated, it is given another name. Only this area is given the name of SLEX. SLEX stand for This is selectin, what is this? Sorry, sialylated Lewis X. Select stand for sialylated Lewis X. So what is sialylated Lewis X? It is tetra, what is this? Tetra oligosaccharide, which is having sialic acid as a part of it. So sialylated Lewis X. And another important thing here is F. It is, don't remember F word, this is for Foucault's. So this sialylated Lewis X or sialylated oligosaccharides or PSGL1 with this area has two important parts. Number one, sialic acid, and number two, Foucault's. Now, this binding, now from the endothelial cell, there is expression of P-selectins, very rapid expression because they were P-selectins were pre-found, pre-stored in viable palate bodies, and at the site of injury, immediately histamine is released and injured endothelial cell can release thrombin, and both of them activate the endothelial cell and force the endothelial cell to release the viable palate body, and P-selectins will express on the endothelial cell. P-selectins make bonding with their counterpart molecule very weakly, calcium dependent bonding, but this is weak and transient bonding. Due to this reason, neutrophil will attach and then blood flow may push the neutrophil forward, then they will detach. So they will just attach and detach, right? We call this process of attach, detach and little bit move on. Attach, detach and move on. This process is called rolling. So here I have explained the role of P-selectins and their counter molecule in what? Rolling, right? And this receptor for P-selectin, what was its name? It has given, the full name of this molecule is PSGL1, that is P-selectin glycoprotein ligand 1, which bind with P-selectins more avidly. It also bind with E-selectins, number one. And this component, the black component, this short component is called oligosaccharides, and they are bound with this red component which is glycoprotein. So we also call this whole molecule sialylated oligosaccharide attached with glycoprotein. Then another name only for this black motif or black component is this, right? Is called SLEX. So in many books you will read that P-selectins or E-selectins, their receptors on the neutrophils or leukocytes are PSGL1, or there are sialylated oligosaccharides attached with mucin-like glycoprotein, or it will be simply mentioned that there will be glycoprotein with SLEX. What is that? Sialylated Lewis X, which is actually a short carbohydrate chain, right, having sialic acid and also Foucault's, right? Now we come to the next point. From where the E-selectin come? We have already discussed about P-selectins. Let's see from where the E-selectin comes. Now, E-selectin does not appear on endothelial cell, even though E-selectin, E for endothelium. E-selectins appear on endothelial cells within 1 to 2 hours. E-selectins do not appear very rapidly, right? So now the question is that why P-selectins takes long time? They take 1 or 2 hours to appear on the endothelial cell, and make the endothelial cell little more adhesive. Right? Now, E-selectins are, from where they come, remember, they are not pre-stored in endothelial cell. P-selectins are pre-stored, P-selectins, but E-selectins are not pre-stored. Actually, interleukin 1 and tumor necrotic factor and from gram-negative organism lipopolysaccharides, they act on the endothelial cells. You know, when injury occurs, mast cells, local resident macrophages or dendritic cells, and even activated endothelial cells, all of them can synthesize and then release interleukin 1 and tumor necrotic factors, which are cytokines, and also lipopolysaccharides, which are derived from gram-negative bacterial membrane, outer membrane, lipopolysaccharide. Now, actually, endothelial cells do have receptors for interleukin 1. They also have receptor for tumor necrotic factors. They do have receptors for, what are these? Lipopolysaccharide. These receptors give signal inside the endothelial cells, right? When interleukin 1 receptor or tumor necrotic factor receptors or lipopolysaccharide receptors present on endothelial cells are activated, they give signals to the cell to activate a very special protein, which is a master key for many genes. This is a master key. This is a master key which can open many genes. It means these interleukin 1 and tumor necrotic factors and lipopolysaccharide, when they act on endothelial cell, they are going to activate their endothelial cell genetic machinery by activating a special master key. This master key is basically a transcriptional factor. Actually, this transcriptional factor, you might have heard of its name. Its name is NF-kappa B. Nuclear factor kappa B. Now, what is this nuclear factor kappa B? Nuclear factor kappa B can be activated in many cells, and this is a pro-inflammatory transcriptional factor. Write it down. It is NF-kappa B is basically pro-inflammatory transcriptional factor. Pro-inflammatory transcriptional factor. Once it is activated, it will go to the nucleus of endothelial cell and it will activate. Yes, it is activate many genes which produce pro-inflammatory peptides and proteins. It means in the zone of inflammation, these endothelial cells not only acted upon by histamine and thrombin and brought preformed P selectins on the surface, rather within 1 to 2 hours, a real action will start from interleukin 1, tumor necrotic factor or lipopolysaccharides, and they will activate the endothelial cells to make them more adhesive. How? That they will activate NF-kappa B, and this is a transcriptional factor which will move to the nucleus and activate many genes, and those genes will produce pro-inflammatory cytokines and also produce chemokines and also produce some adhesion molecules, right? Right now, we are focusing on one adhesion molecule, that is E-selectin. So E-selectin will be synthesized and then expressed on the surface of endothelial cells. And this E-selectin, because it was not preformed, it cannot come immediately after injury. It will take 1 to 2 hours that endothelium in injured tissue will become expressive of E-selectin, right? Now, once the E-selectin come over here, right, again similar receptors, there are, what are those counter receptors on here? Yes, you remember that. What were they? PSGL1, which were also active on the P-selectin. They also work on E-selectin. Or we can call them sialylated oligosaccharides attached with mucin-like glycoprotein on leukocyte or neutrophils. Or we can call it that glycoproteins on the, what is this, neutrophil will be having, what is that, SLEX, right? Selectin, what is that? Sialylated Lewis X, sialylated Lewis X, right? So the same molecule which was binding with P-selectin will be also now binding with E-selectin. But one thing is there, you know, E-selectin bind it little more strongly. I think E-selectin is too good in kissing. We can say P-selectin and its receptors were undergoing soft kissing and moving on, soft kissing and moving on, you know, soft kissing, lip to lip, but with closed mouth. Here, French kiss is going on. What will happen? Endothelial cells are more activated. Now leukocytes are more activated and they become more adherent, and rolling further slows down. So we can say rolling is here being mediated by P-selectins and E-selectins, which are now being expressed on endothelial cells, and their receptors which are being expressed on neutrophils. But E-selectin, very weak binding, very transient. Sorry, P-selectin. P-selectin binding appears earlier but very weak and very transient. E-selectin is moderate but it appears late. And after 1-2 hours, endothelium in the area of inflammation is loaded with P-selectins as well as E-selectins, and leukocytes are also expressing sialylated oligosaccharide with their glycoproteins attached with neutrophil membranes, and they are attaching, detaching with each other. So what happens? Here was initial rolling, which was P-selectin dependent, then E-selectin dependent. After 1 to 2 hours, stronger attachment, not very strong, not hugging, just kissing, still kissing, but with open mouth, with little bit tongue action. I mean, probably they are trying to burn some calories, right? So P-selectin interaction is like soft kissing. E-selectin interaction is like French kissing, right? And now neutrophil, when E-selectin dependent interaction occurs, neutrophil and endothelial cell become more active. And when they become more active, they are now getting ready to hug each other by activating their integrins and expressing integrin receptors here. That we will discuss in next lecture. But before I finish the lecture, I want to mention one thing about L-selectin. L-selectins are L-selectins are normally expressed on microvilli of, what is this? Leukocyte, especially on lymphocyte. L-selectins normally appear on leukocytes, not on endothelial cell. You know, P-selectin was present in platelets and wobble palette bodies of endothelial cell. E-selectins were induced, synthesized, and then expressed on endothelial cell. L-selectins are present on many leukocytes, including lymphocytes. L-selectin has a very special role. Receptor for L-selectins are specially present in lymph nodes. So when lymphocytes go to the lymph node, in lymph node there are some special venules which are having not flat endothelium, rather little tall endothelium. Those are called, those venules in, what is that? Lymph node, right? Those venules are called high epithelial or endothelial, high endothelial venules in lymph nodes. Lymph nodes. So high endothelial venules are present in lymph node. These venules express the receptors for L-selectins. So when lymphocytes are moving through the lymph nodes, when they reach this area, they will become adherent, and then lymphocyte will extravasate and go inside the tissue of the lymph node, and there they will meet with the antigen presenting cells, right? So actually we say L-selectin basically are responsible to manage the lymphocyte traffic through the lymphocytes and pair patches. Pairs patches are in gastrointestinal tissue, right? Wall. Pairs patches and lymphocytes, especially in the follicle, there are special type of venules which are called high endothelial venules, and these high epithelial or endothelial venules, right, they specially express the receptors for L-selectins. And here it is very important to remember that many lymphocytes express the L-selectins on their microvilli, and through the L-selectin they bind in the lymphocyte special receptors in the lymph nodes and pairs patches, and we can say L-selectin is very important for trafficking of what? Trafficking of movement of, yes, lymphocytes, right? Now you have already seen that what is rolling and how it depends on selectins. Remember, rolling is a reversible process. If after a lot of kissing, hugging does not occur, neutrophil will just slip away. It means this soft kissing and then French kissing should be followed by a strong bonding between the neutrophil and the endothelial cell, which is integrin dependent interaction. We call it firm adhesion. If integrins are defective and firm adhesion is not there, neutrophils come, just roll a little bit, and if they are not really caught and hugged by these endothelial cells, then rather than going out, extravasate, they will be taken by the blood flow outside the inflamed area. So it means that it is very important that rolling, which is selectin dependent, should be followed by firm adhesion, which is integrins dependent. That we will be discussing in next lecture. But here I want to highlight a disease. Why I especially mentioned about Foucault's. You remember I was talking about Foucault's. There is a group of disorders which are called leukocyte adhesion molecule deficiency, LAD. LAD is a group of disorders in which there is leukocyte adhesion disorders are there. There's problem with the adhesion of leukocyte. Now, there's a special type of LAD which is called LAD type 2. What is the problem in this? In this disorder, the enzyme which help the fucose to bind, the enzyme which help the fucose to bind the P and E selectin receptors, right? That enzyme is either not produced or it is defective. It means fucosylation of SELEX is not done. That enzyme is called fucosyl transferase. What is it called? Fucosyl transferase. Now, what happens in LAD2, leukocyte adhesion disorder or deficiency type 2, fucosyl transferase enzyme is defective or it is absent in the neutrophil. It means neutrophil will produce SELEX which are defective, they are not having fucosylation, or we can say the, what is this whole molecule called, yes, P selectin glycoprotein ligand, glycoprotein ligand 1. This will be defected. This is an inherited disorder, very rare, that if your genes which are responsible to make fucosyl transferase, those genes are absent or defective, both alleles, there's severe deficiency of fucosyl transferase, then fucose cannot be bound with SELEX or cannot be bound with sialylated Lewis X. So sialylated Lewis X will be defective, or you can say PSGL1 will be affected. In this case, what will happen? Neutrophil come here, marginate, but they will not kiss. They will not bind with P selectins and even they will not bind properly with E selectins. So they will not undergo rolling. If they don't undergo rolling, these leukocytes, even though they are marginated, but they don't become slow. Because purpose of rolling was to attach, detach, make it slow. So neutrophils come on the margin in the inflamed area, but still very rapidly are washed away or moved away due to blood flow without attaching properly to the endothelial cell, right? They will go back by the venous blood. What will happen? That leukocyte, if they are having defective rolling, here there is what? Defective rolling. Rolling is not going on. So if rolling is not going on, even integrin dependent hugging can be done, but neutrophils are not having initial attachment. So neutrophil will go away. So what will happen in these? The neutrophils will not be able to extravasate. They will not be able to roll. So of course there will be no significant firm adhesion, no significant diapedesis, and no pus formation, and what will happen? Very severe and very recurrent infections, right? In LAD2, there will be very severe and very recurrent infections. Why? Because fucosyl transferase deficiency result in defective receptors for P selectin and E selectins, so there is no rolling step and neutrophils cannot bind properly with the endothelial lining, and even when there is no rolling, that translates into minimal firm adhesion and minimal diapedesis and eventually failure of formation of even pus and failure of clearance of pyogenic organisms. So bacterial infections are very common in these children, and along with that they have one more issue, that you can say that in the blood total leukocyte count goes high due to repeated infection, plus because neutrophils are not going to fight, they are just circulating, and the cytokines from inflammatory area which are being produced, they are increasing the leukopoiesis, and they are keeping the bone marrow marginated or bone marrow stores of neutrophils very active and released. So what happens in these patients? These are the patients which have children which have repeated infections, severe infections, pyogenic infections, no pus formation, but total leukocyte count in the blood is very high. So we can say neutrophils are there, but they are unable to roll and firm adhesion and diapedesis, right? And all these problems will lead to, you can say, very much reduced life expectancy. Another thing which is common in these patients led to is Bombay blood group, right? I will explain it in some other time, but in Bombay blood group there is H antigen and A antigen and B antigen, none of them is present on the red blood cells, right? So their blood group is not A, not B, and even H antigen is absent, so their blood group is not O, right? But these patients with Bombay, you know, normally on red blood cell what happens? Suppose these are 3 red blood cells from different patient I show. This is H antigen, H antigen, H antigen. Here H antigen is modified into, here H antigen is modified into A antigen. So blood group A. Here H antigen is modified into B antigen, right? So blood group B. It is very simple explanation. Otherwise I have a very detailed lecture on ABO blood grouping. And third is, if H antigen is expressed but there is no modification of it, then we say it is blood group O. If both of them are present, for example, some of the H are converted into A and some of the H antigens are converted into B, then it is blood group AB, right? But if there is red blood cell, right, and there is no H, this H is not there, so of course this cannot have A group, cannot have B group, and cannot have O group. This is very rare group, blood group Bombay blood group. And this Bombay blood group, we say that genes to make H antigens are absent on both allele, right? So it will be HH, small h, right? Not capital H, which shows the presence of the gene, functional gene. Now, Bombay blood group, what happened? When you have blood group A, in the plasma you have anti-B antibody. I have explained in the lecture in ABO blood group how it happens. If you have blood group B, patient will have in its plasma antibody anti-A. And if you have blood group A and B both, AB, you will not have anti-A, you will not have anti-B. Is that right? And if you have blood group H, it means you don't have A antigen and you don't have B antigen, but your plasma will have anti-A and anti-B. But if you go to this Bombay, because there is no H antigen, this will have anti-H. Of course, no A antigen, so it will have anti-A. And no B antigen, so it will have anti-B. So what happened? Bombay blood group is characterized by absence of antigens H and A and B. All 3 of them are absent from red cell membrane. And plasma has all 3 antibodies, anti-H, anti-A, anti-B. This Bombay blood group is also associated in the patient with led to, because that defect is also occurring from fucosyl transferase deficiency. Right? So here we stop today, and next lecture we'll discuss into detail about, yeah, romantic business was going on, love affair was going on between neutrophil and the endothelial cell after soft kissing, which was p-selectin dependent, and French kissing, which was e-selectin dependent. Now is the time for firm adhesion hugging, which is integrin mediated. That we will discuss in detail next time. That what are integrin molecules, what are their ligands, and what diseases occur when integrin molecules are defective. Right? Thank you very much. See you later. Please like, subscribe and share this video, and don't forget, I love you all.